Hello everyone, welcome to a brand new video on thyroid hormone production and secretion. So in this video we're going to discuss the thyroid follicle, its structure. We're going to look at how it's stimulated through TSH. We're going to look at how T3 and T4 is produced with the 10 steps in that production. So without further ado, let's jump into it and discuss first off our thyroid follicle. Now the follicle is going to be the functional unit of the thyroid gland for this particular thing. And it's called a thyroid follicle. It is the particular functional unit. So it's got a structure made up of a very large kind of duct made up of a lining of simple cuboidal epithelium. And what we want to make sure we understand about this follicle is basically its overall arrangement and how it associates to other things. Now, there are other cells that are going to be around here that we're not going to talk about today. I might get into a little bit in another video, but let's just talk about this thyroid follicle, this simple cuboidal epithelium wall that we have. And this wall will be in proximity to a capillary, series of capillary beds. So let's put a capillary present as well. And let's label one important area, the follicle lumen. This will be the particular important thing. Sorry, I was trying to write in the follicle lumen. And what I want to do is discuss that this follicle lumen and one of these follicular cells here, that's what we're going to focus ourselves on. So in this video, I want to focus on one of these follicular cells. Let's say one of these here near the capillary bed, and let's just discuss that and see what happens in a little bit. Now, as we also want to do is remind ourselves of the signaling that takes place in here. And to do that, let's say I've got one of these follicular cells right here. Here I have a follicular cell at the bottom of the thyroid. The circle is going to be the anterior pituitary. So let's write A, P for anterior pituitary. And then the big oval is going to be the hypothalamus. And let's just write hypo, period, for the hypothalamus. And here at the bottom we have a follicular cell. Now what we're going to do is we're going to release a hormone from the thyroid called thyroid regulating hormone or thyroid releasing hormone, TRH. We're going to be able to regulate the anterior pituitary to then release a hormone called thyroid stimulating hormone. And we're going to look at how that thyroid stimulating hormone will go into this to cause this follicular cell to ultimately produce the hormones of T3, triiodothyronine, and T4, tetraiodothyronine, and how we're going to be able to use that to produce those hormones. Now, just for your edification, if you have once this level goes up, we can use a negative feedback mechanism to turn that off and stop producing this hormone. And this is how we regulate through negative feedback the hormones production. So, what we want to focus on is this hormone production of T3, T4, and looking at a follicular cell. Now, I am not going to go through and draw every piece of detail that I can about a follicular cell. I just want to make sure we understand this is a follicular cell right here. This is one of those follicular cells that we talked about in the follicle of the thyroid. This will be the side where the capillary's blood is. And this will be the side with the lumen. And the thyroid follicle. Okay, so what we want to talk about is starting off, let's make sure we just bring in and discuss a little bit of our hormone signaling that we discussed. And let's say I have a receptor right here, and this receptor is going to bond to TSH. So TSH comes in, the hormone, thyroid stimulating hormone, TSH, will bind to this. Now, because thyroid stimulating hormone is a peptide hormone, it will bind to it and it will activate a second messenger system. In this case, it will be cyclic adenosine monophosphate, or CAMP. Now, CAMP system activates ultimately various protein kinases. This will be protein kinase A, PKA. And what this PKA will do is it will act as a transcription factor. Sitting inside the nucleus of this cell will be some DNA. And on this DNA, there will be a gene that will code for a particular protein we want to make. And this PKA will come into it and bind to it to stimulate its production. Now, this gene will get transcribed. Its mRNA will leave the nucleus. It will go on to the ribosomes of the rough endoplasmic reticulum, for example, and be read, be translated into its protein. And this protein that we're going to make, let me put our rough ER, and I'm just going to call it RER, rough endoplasmic reticulum. And it will produce a protein here, and this protein is called thyroglobulin. Now, I'm just going to put TG for short, thyroglobulin. And what we're going to do with thyroglobulin is, let's say we have this kind of curly tail, and then I'm going to put 4 little rings on this thyroglobulin sticking out. And these are going to be our tyrosine residues, which are going to be important. The tyrosine residues are going to be important to the production of this hormone. So this is thyroglobulin. Now, what we're going to do with thyroglobulin is we're going to release thyroglobulin out into the lumen through exocytosis. Let's see, let's draw this guy out. Now, I'm going to make him a little bit bigger here, and I'm going to put 4 little rings. Try to draw 4 little rings, kind of pretty here. And these 4 little rings are going to be very important for us momentarily. These are those tyrosine residues that are on, those are the amino acid tyrosines, the R group residues that we're going to be needing so much to make this hormone. Now, what we want to do is we need this thyroglobulin, TG, to be an important component to produce T3, T4. Now, T3, T4, remember I said, is triiodothyronine and tetraiodothyronine. So this is going to let us know that an ion called iodide ion, I- is going to be incredibly important. So we're going to need to get this into the follicular cell. Now, the way we do this is how you're going to get a lot of iodine into cells is we have a particular type of protein here. Now, I'm going to draw 2 of these. I'm going to put this one first, and this is going to be the Na plus /I- Symporter. Remember, Symport or co-transport is the transport of 2 substances at the same time in the same direction across cell membrane. Now, how are we going to do this? Well, if you guys should know by now that there is a protein that all cells have called the sodium-potassium ATPase pump. And that the sodium-potassium ATPase pump, for every molecule of ATP, brings 2 potassium cations into the cell and pumps out 3 sodium cations. So sodium is always very high. There will always be a lot of sodium in the ECF. In the plasma, it's about 135 to 145 milliequivalent, so it's quite high. So what it's going to do is it's going to want to follow its electrochemical gradient and come in to the cell, this Na+. Now, with that, we're going to have a piggyback writer of I minus who wants to piggyback off of that and also come in to the cell is I minus. Now this I minus is piggybacking off of this. Now this makes the sodium iodide Symporter a secondary active transport protein, whereas the sodium potassium ATPase pump is primary active transport. That is the most important primary active transport protein you could ever know in your education, okay? Now this iodine has been brought in, and we're going to have one more important transport protein here called pendrin. And this pendrin protein is going to take this iodide ion and secrete it, help it get into the follicles lumen. Okay, and so it's going to carry it into the follicular lumen. Okay, now once we got that in the follicle lumen, we're going to be able to take this iodine now and stick it to our tyrosine residues. But something's got to happen to this first. And what's going to happen is there is an enzyme here, this enzyme that we have that's going to be able to help take this thing is thyroid peroxidase. We need to take the enzyme thyroid peroxidase, TP, and we're going to convert this iodide into an iodine or into an iodine ion, cation, and we're going to be able to do this. This is called oxidation of the iodide. Oxidation reaction. Remember, loss of electrons is oxidation. Anytime that you are going to lose electrons, you are oxidizing something. Now that we have oxidized this, these, especially the elemental iodine, is now reactive. It's going to be quite reactive. And what we're going to be able to do with this is we're going to be able to stick it to the thyroglobulin. This thyroglobulin, as it comes by, we're going to be able to stick this iodine onto these tyrosine residues. So let's do that. Okay, so let's take some of these iodine residues. And let me be really good and careful with these so they look pretty as I'm drawing it. And what I want to do is especially as I iodinate them, let's stick an iodine. Let's stick one iodine on this one. Let's stick two on that one. Let's stick two on this one. And let's stick two iodines on this. This is called iodination. We are iodinating the tyrosine residues. Some are getting only one iodine, some are getting two. Now, what is about to happen is something very fascinating. We're going to take this iodinated thyroglobulin, and we're going to do a little rearrangement. Okay, we're going to do some rearranging of this, and we're going to rearrange it a little bit, which is one of the most fascinating processes. We're going to take this one from here and stick it on. Now, make some special note here because this is very important. Notice that this iodine residue, this iodine here, there was only, on that tyrosine residue, there was only one iodine, and this one has two. Okay. Now, what we're going to do with the other one here is, we're going to take, let's say, one from here and stick it on this one. Okay. So we're going to have, again, two tyrosine residues, and we're just going to shuffle things around where we're going to have two iodines on it and two iodines on that one. Okay, so this is very crucial, guys. This is a really crucial step into producing the hormones T3, T4. We're really close to that now. So we have done a process that is called conjugation, the rearrangement here of these residues. And these iodinated residues, we have now done a conjugation of them. So what we're going to do is pick this up through endocytosis and bring this in during a vesicle here. We're going to bring a vesicle in with these iodine, with these tyrosine residues on it who have been iodinated, and we're going to bring them in. Now, what I want to do is to make sure that I get these iodine residues visible for us. We have some with 3 and some with 4 iodine residues on it. Now what is going to happen is we're going to take that curlicue part off that I had, the big tail, the thyroglobulin, and we're going to leave behind just these tyrosine residues that have been clipped off in a process called proteolysis. Proteolysis. And while we have our complete our hormones, this is now both T3 and T4. I have my T3. Notice there are 3 iodines on the tyrosine residues, and this one here is T4. So now all I've got to be able to do with this T3, T4 is secrete it. We're going to use a protein called a monocarboxylate transporter, an MCT. Let me erase that. I meant to use a different color here just for some purposes. I was going to use a green just to make it stand out. All right, and we're going to secrete this out. These secreted out here is our T3 and our T4. Okay. Now I'm using circles for the rings because it is easier to draw the circles than it is to actually draw the benzenes and things like that. Now, one of the cool things is when they get out into the bloodstream, they're going to ultimately bind to a thyroid binding globulin to actually transport it. And so let me signify that here. Is that these guys can bind to thyroid binding globulins to carry it through the bloodstream and get it to where it needs to go. So a lot going on here, very complex process, but I hope we broke it down step by step and explained it in a way that made this understandable to you guys. And always one of my favorite things to talk about is endocrinology. I enjoy teaching it. I always love it. It's complex, and that's why I love it. I love the complicated things the most, it seems. So guys, this concludes my video on thyroid production and secretion, thyroid hormone, T3, T4. I hope you found this video helpful. If you would like to see other videos in the future, let me know what you would like to see. But this concludes everything for this video, and I'll see you guys in the next one. Thank you so much, and thanks for watching.